Well, welcome everybody. Thanks for coming out for this talk on understanding ethylene oxide sterilization. Just by a quick show of hands, how many people currently use ethylene oxide? So we have a few, and how many have never used it? So good. So we have a good mixture of the two. So what we'll do is we'll talk a little bit about the introduction, some of the history of ethylene oxide, some of the benefits of it, how it works. Then we'll get into how we design a cycle. When you have a product, you first have to design it with sterilization in mind. You want to find out, can this product be sterilized with ethylene oxide? What are the challenges to that? And then once you go into that, find out what cycle you need. Then you go into the validation part of it. And then finally, we'll talk about some recent changes to it, so the standards. So as an introduction, some of the advantages of ethylene oxide. Ethylene oxide, just as a background, it's used for about 56% of medical devices that are sterilized in a large industrial scale. So the other approximately half is done with radiation. So ethylene oxide radiation are the two main ways that you sterilize on a large scale. Ethylene oxide's real niche is low temperature, so a lot of the plastic devices, polymers, things like that can't withstand high temperature like steam or dry heat, or radiation material compatibility is going to be an issue with radiation, so ethylene oxide is very gentle on products, so that's another advantage for that. Another advantage is it's considered an overkill process, which is, in a way, it's almost an advantage and a disadvantage. The advantage is that it gives you a lot of safety factor built into your process, so you're able to withstand larger fluctuations in your bioburden. If you look at a process like radiation, if you have a spike in your bioburden, your dose is set directly on exactly what your bioburden is. And so if you have a spike in your bioburden, that can affect your sterilization validation that you've done. With ethylene oxide, there's so much overkill built into it that it's going to allow you to withstand larger swings in that without having any issues with it. It can be a little bit of a disadvantage because it is an overkill process. Your cycle is going to be longer than it really needs to just kill the product. There's a big safety factor built in. Penetration. Penetration is ethylene oxide's main advantage. You look at things like vaporized hydrogen peroxide. On paper, you would think that would be a better sterilant, because with vaporized hydrogen peroxide, you're sterilizing in the range of like 1.2 milligrams per liter. With ethylene oxide, around 650 milligrams per liter. So you would think that vaporized hydrogen peroxide would be a more powerful sterilant. But the problem with that is it can't get into the device, to the areas on the device that it needs to get to be sterilized. And so with ethylene oxide, it's such a small molecule, it's very penetrating. You'll actually penetrate through the walls of plastic, so that's one of its biggest advantages. Regulatory acceptance is another one. It's been around for approximately 60, 70 years, using it the way we use it today. Everybody's very familiar with it. The FDA is familiar with it, regulatory bodies. So if you're using an ethylene oxide process, it's something people are used to seeing, and there's going to be a lot less questions than if you were trying to use a new novel approach. The revalidation interval, that's an advantage as well. You only have to revalidate by doing an actual physical run once every 2 years. In those off years, you can do just a paper justification as long as nothing's changed to the process, as long as you haven't had any failures, non-conformances, or problems with your process in that last year. You just assess the process and go through, and then you can decide if you need to do a physical run or not. Also used for small or large volumes. Small is just a couple of boxes, all the way up to some chambers hold more than 28 pallets, a full tractor trailer, so a wide range of flexibility for that. A little bit of history. Again, it's been around for a long time. In the 1920s, they first used it as an insecticide. This is a picture of inside a grain elevator. They used it a lot for grains and cereals and things like that to kill insects. And so they would put ethylene oxide in with dry ice and allow that to kind of fumigate the grain elevators. So in the 1930s was the first time they used it to kill microorganisms. So kind of moving towards an approach like we use today, where we're killing bacteria, viruses, molds, things like that. And so that was in the late 1930s. In the 1940s, they first started using ethylene oxide for food, to preserve foods. They developed a cycle that was similar to what we use today. And if anyone's heard of Sterigenics, most of the people that deal with ethylene oxide have heard of Sterigenics. Before that, they went through some name changes. It started out with Griffith Microscience, and then before that it was Griffith Laboratories. And this C.L. Griffith, he was one of the ones that first started using this. Again, they used it not for medical devices at this point, they were using it to sterilize foods. And then along came World War II, there's a big need by the US government to get large quantities of food shipped over to Europe and to maintain that to feed the troops. So this is where they developed sea rations, where they canned a lot of food and preserved it. So they spent a lot of time and effort using ethylene oxide to extend the shelf life of food. A lot of the work by the government they did to optimize the parameters. A lot of the parameters that you use with ethylene oxide, it's very complex. You have temperature, gas concentration, relative humidity, and exposure time. So all of those 4 parameters have to work in conjunction together to be effective. And so there was a lot of research on what happens if you change just the humidity or just the temperature. And then really with a lot of the information we use today, like a lot of things came out of the US government working on it. And so we still use that today. Kind of some funny looking picture of an old sterilizer, pretty rudimentary. They used 55-gallon drums when they first started out. They'd put whatever materials they wanted to sterilize it. They used these cans that had a valve on there that they could open the valve, allow the ethylene oxide to go into this barrel basically, and then let it sit for a specified amount of time, and then whatever was in there is sterile. Obviously today, much more complex. If you stop by 3M's booth, they have one of their sterilizers, same one that's in that picture, that has a lot of functions as far as programmability. You can program almost anything you want to, how fast the gas adds, how fast the vacuums go. So all computer controlled, and so very, very obviously much more complex, and the process has really been optimized a lot since those early days. If you're using ethylene oxide sterilization, you really have to understand the overkill method. For all types of sterilization, except for radiation, you use overkill processes. And that's basically the concept of you're using a highly resistant biological indicator that you put in the hardest part of your product, and you got to show that you kill that in half the time. So then your routine cycle is going to be double that exposure time. So that's the amount of overkill that we told you is built in. So you find the time that it takes to kill 1 million of the hardest to sterilize organisms in the hardest part of the product, and then you're going to double that processing time. So really the cycle is twice as long as it needs to be, but that's really where you get that flexibility and that process. A lot of you have probably heard the term sterility assurance level of 10 to the minus 6. That's basically 1 unit in a million being non-sterile. And so how we do that is we use a biological indicator that has 10 to the 6th or 1 million spores, and by killing that in half the time, that's where you're going to show that process, because then you're going to assume it's considered to be first order kinetics that you get the same kill rate in the second half of the cycle as you did in the first half of the cycle. And so by going through this, if you show a 6 log reduction in one half of the cycle, when you do the other half, you're going to get another 6 log reduction. And so that's going to take you down to the 10 to the minus 6. And that's what the requirement for terminally sterilized devices are for. Okay, we'll get into that in just a sec. The question was, how long does it take to sterilize? In just a second, we'll show you how we go through and actually determine how long. The answer for that is really depends on the product and the load itself. It varies from a lot of different times. So when you're looking at ethylene oxide, again, we say we use these indicators that we put inside the product. What we use is a spore call. It's an organism that forms a spore. A spore is kind of like a seed is to a plant. It has a protective coat around it that's very resistant to chemical attack. It's resistant to heat. And so it's very difficult to kill these organisms. We're in that spore state. So this is called an organism called Bacillus atrophius. It's the indicator for ethylene oxide as well as dry heat sterilization. Every type of sterilization has its own indicator organism. Steam sterilization uses an organism called Geobacillus stereothermophilus. So with ethylene oxide, we use this particular one. It's a gram-positive spore-forming organism. Nice thing about it, it has orange pigmentation. So when you do the testing on it, it's very easy to see, just visually, even if you have what's called a true positive, or if it's a contaminant from the testing process. So it makes it very easy to work with. So why do we use bacterial spores? A lot of times people will ask, well, how can we use just this one organism, but there might be viruses, there might be molds, there might be all these other types of microbes on my product? And the answer is that bacterial endospores are the most resistant entity to this type of sterilization. Again, because they are a spore former, it's going to have that protective coat around that. And so the thought is you kill bacterial endospores, all these other organisms are so much less resistant that you're going to kill those as well. That's why when you do a sterility test, you don't have to test for viruses, for protozoa and things like that might possibly be on there. Viruses are quite easily killed very easily killed with ethylene oxide. So that's why we use bacterial spores. That's kind of a key to ethylene oxide sterilization. So we take that organism and we put it on what they call a biological indicator. Typically it's just a paper strip that has the spores of that bacillus organism. They put a liquid suspension onto that, allow it to dry, and then they package that up into a biological indicator. And it basically only has that one specific organism, and then that's placed inside the product, inside the load, and that's how you monitor sterilization. After you sterilize the product, you pull those out, you test them, and you're looking for either growth or no growth. And so that's what a biological indicator is. But the reason you can use just this biological indicator and not have to test your actual product is a lot of times you'll develop what's called a process challenge device. This is something that if your product is very expensive, or it's something that's hard to get that biological indicator in the hardest part to sterilize, you'll design something that mimics the resistance of that, but you don't have to use your actual product. Something that might be very expensive. Some people have a product that's over $1,000 a unit, so if you had to waste 20 of those samples just to put biological indicators in, that's a lot of money, where you could maybe use something like a 5-cent syringe or something very cheap and easy to work with. So process challenge devices are very commonly used as well. So now we get into the cycle and PCD development. This is where we get into how long your sterilization cycle may need to be. When you look at your product, you got to assess all the different areas on that device that might be hard to sterilize. Things like stopcocks or closure systems, anything that has caps that close off the device, that's going to restrict the gas getting into that part of the device. These are areas that we're going to challenge on the product. If you have lumens, long, narrow lumens, again, somewhere where the gas is going to have to get down into that hard to sterilize part of the product. These are all things you have to look at and determine if you can sterilize it with ethylene oxide or not. Mated surfaces is another one. That's one where two surfaces are pressed up against each other. Again, it's a barrier to get the gas down through that, although it will penetrate through the walls of that syringe as well as through the plunger itself, so it is very penetrating. But these are all things that you're going to look at. Sometimes the packaging itself can be a barrier to sterilization. So typically you're going to use something similar to Tyvek or paper. Most people use Tyvek as a material that's a microbial barrier, but it's also breathable and permeable so that the gas can get into and get back out of it again. So the thing with ethylene oxide sterilization, there's a number of vacuum and pressure changes. So you'll pull a deep vacuum, you'll allow air to come back in, you'll do that multiple times because at the end of the cycle, you have to get all of that ethylene oxide back out of the product. So you're going to pull multiple vacuums, so the pressure changes, the package has to be able to withstand that. If anyone's ever been camping or driven up in the mountains with a bag of potato chips, the bag expands and almost explodes because the internal pressure, as you go higher, the atmospheric pressure is lower, so that bag is the pressure inside can't equalize with the outside, so the bag swells up. Same thing with ethylene oxide. When you pull a vacuum on the product, the packaging needs to have some way to equilibrate that pressure, or you can end up bursting your seals. And so that's why all types of packaging for ethylene oxide has to have some type of breathable area to allow that to occur. Foil pouches is an example of a foil pouch would not be a good candidate for ethylene oxide sterilization unless you had some kind of Tyvek header, or sometimes they have little patches of Tyvek that allow the gas to get in and back out again. So that's the packaging. So once now we've taken our product, we've looked at the different areas that we're going to sterilize, and then we have to inoculate those. And we have to test every one of those locations to make sure that the gas is going to be able to get in there and kill that location. FDA is will commonly scrutinize where you inoculated. So what we'll tend to do is we'll inoculate multiple sites along the device. And then as you go through and do the testing, the easy ones will drop out. You're going to do a series of fractional cycles. And so the goal is you're going to try to find out what's the last site on the product that's going to survive. And again, when we looked at all those different parts of those devices, sometimes you can't get one of those big paper strips into the hardest part of the device. If you have a small, narrow lumen, you might have to use something like this. This is where we inoculate surgical sutures with the liquid spore suspension. So we're putting the spores on in an ethanol-based solution. That ethanol then flashes off, it evaporates off, leaving just the spores onto that suture. Then you can place that suture down inside that narrow lumen. Or sometimes you use the liquid alone and put that on one of those mated surfaces where they're going to be pressed up against each other. So you'll put it on that surface, allow the spores to dry on there, and then reassemble the device. So sometimes you have to get very creative on how you're going to get the spores in the hardest part of the product. That becomes one of the more difficult things. And I mentioned we use process challenge devices a lot because you want to come up with something that you can use to monitor the cycle so you don't have to use the actual product. So we'll typically add a whole lot of different ones. We don't know which one's going to work at the beginning. Usually we have a fairly good guess. So we'll try something from low resistance, medium, and then high resistance to try to find one that's going to most closely simulate the product. Because what the standard state is, the process challenge device that you use has to be at least equal to your product, if not harder to sterilize than your product. And so generally, we'll try all these different ones. We'll put those process challenge devices that we're going to try. We don't know which one's going to work, so we'll try several different ones, and we'll mix those evenly in with the product because we're doing a direct comparison of two different items. We want to compare the product to that process challenge device. And again, the product is going to be inoculated at multiple locations, all these different sites. And then we're going to run fractional sterilization cycles. So we're going to do a run, maybe we do it 30 minutes, then 60 minutes, then 90 minutes. After each run, we pull those samples out, test them, and we're looking to see if anything survives. So you're just doing a regular sterility test of your biological indicators. It's a 7-day test. This is a little different from the product sterility test when you're testing your actual product that goes 14 days. This is going to test for 7 days, and you're just looking for growth or no growth. It's a plus or minus test. And so when you do that, you tabulate all the results. In this case, we did a study where we ranged from 15 minutes, 30, 60, 90, 120, and 150 minutes, tested all these different areas. In this case, it was a catheter type product. So we tested multiple areas across the lumen of the middle, the beginning, the middle, and the closed off end of the catheter. We tested any of those mated surfaces. And then if you see those last three things called, those are the different process challenge devices that we use. So again, we try different ones, we're not sure what's going to work. And so you're trying to find one that's equal to or greater than the product. So if you look at the 60-minute column there, all the product sites are killed off. So that's the endpoint for the product. And so you're trying to assess which one of those three PCDs is going to be equal to or outlive the product. And as you can see, all three of those at least survived as much as the product, if not more. So any one of those could really be used to monitor your process with. Does anybody see a problem with that first one that has growth all the way across? We don't know when that dies off. I mean, we go all the way out to 150 minutes and it's still alive. Problem is it may go all the way out to here, maybe hours and hours to kill that. So you always want to make sure you find an endpoint so you make sure you don't come up with something that's too resistant. And so any one of these can be used for your product. You just want to pick the one that's most appropriate. And sometimes people will choose ones that are way more difficult than their actual product. And what that'll do is that'll give you more flexibility down the road. If you design a new harder product than this particular product, you can adopt that into your sterilization process without having to go through and totally revalidate that. And so it makes it easier. The downside is you may have a longer cycle than you would otherwise need. So it's a little bit of a balance of how much overkill do you want in to be able to add new products down the road versus how short do I want to keep that process and how optimized do I want it. So does anybody have some ideas where you would inoculate this product? This is just one that we've come up with. We put a piece of tubing on a syringe. Does anybody see any locations on there that would be concerning to you? Sorry? Yep, where the plunger is, that's a mated surface there. You also have in the barrel of the syringe itself, the gas has got to get all the way down there. That cap on the end, whether it's vented or non-vented, could be a big deal. If it's vented, it's going to allow the gas to get into that more easily than if it's non-vented. If that's a non-vented cap on that, then the gas is going to actually have to penetrate through the walls of the syringe or the tubing or the backside of that plunger to get in there. So you would inoculate most likely along the lumen, under the cap, inside the syringe, and then possibly even on the threads of where that lure lock screws onto that, because that's going to be a mated surface as well. And so as you can see, even if you have something that looks fairly simple, and a lot of devices are way more complex than this, that even with this you would have multiple inoculation sites. Sometimes we'll get kits that have maybe 50 or 60 components in there, and you have to try to go through all of those components to try to find what's the most difficult to sterilize. So the next test that we do before we do the validation is bioburden enumeration. This is a study where we're going to find out how many microorganisms are on the device just through the manufacturing process. after it's gone through all the manufacturing steps, up to but not including sterilization. Sometimes we get people that will send in samples to test for bioburden that have been sterilized. Obviously, if you do that, your bioburden is going to be very low because it's sterile. So this is a test you do on non-sterile product, but you want it to be representative of going through every step that you do, because every time you handle the device, you have organisms that just fall out from the air and settle on the surface of the device. If you have any water steps where maybe there's a cleaning process, if that's allowed to sit wet, a lot of microorganisms, if there's water present, they will start to reproduce and can actually increase on the product. So water is always a red flag. A lot of the organisms that live in water can also cause endotoxin problems. And so bioburden is a step that you do with ethylene oxide sterilization, just like you do with all other types of sterilization. But it's not quite as critical as it is with radiation. With radiation, you're taking the results from this test and setting your cycle on that. With ethylene oxide, you're not setting your cycle length based on just that number that you're going to get, but it's going to be a judge of your manufacturing process. It's going to show you how clean that product is, or what the bioload on that is when it's ready to go into sterilization. And that's something that's normally done quarterly. You're going to repeat that quarterly, and you'll see that changes over time. As you add new people to your clean rooms, new raw materials, maybe you change vendors, all of that can affect your bioburden. You'll see it go up and down, even with the seasons. When it's warmer and wetter out, more humidity in there, you'll see higher bioburden levels than maybe in the winter when it's colder and drier. And so all these can affect that. That's why you monitor. It's a test you have to keep doing just to see what that bioload on your product is. Another test we do, which is similar to the comparative resistance, in that test we actually inoculated the spores into your device. This one we just want to find out, of all these things that are on the device through that handling and through the environment, are any of these things going to be overly resistant? Anything that has extreme resistance to ethylene oxide? Because if you remember, when we're monitoring the cycles, we're testing that biological indicator that just has that spore strip on there. And so this is where we tie that together to determine that whatever's on that strip, we compare it to whatever happens to be on your device from your manufacturing process. And we have to show that that process challenge device is even more, not only more resistant than when you put the spores in the hardest part of the device, but even more resistant than when you whatever just happens to be on your product. Sometimes organisms can mutate and change and become extremely resistant. So this is a screening test that you do periodically to make sure you don't have any of these strange microorganisms on your product that can cause problems. So again, how you do it, it's going to be very similar to what we did when we inoculated the product. You're going to just test your actual devices. We're not going to even open it up at all. We're just going to take the devices that are all packaged, place them in the sterilizer next to that process challenge device that we came up with, and we're going to do these same type of fractional sterilization cycles, expose them to that, then we're going to do a test on it. In this case, we do an actual sterility test on the product, where we're going to test the product on one hand and then that PCD on the other, and we're just trying to show that whatever happens to be on the device, whether you have just 100 organisms on there, 1,000 or maybe 10,000, that whatever's on there is not going to be more resistant than that biological indicator. So it's a little bit different take on that. So now that we've done all this pre-testing, this is all the stuff that we've gone up to the point we're ready to now go to a contract sterilizer and then validate the process. So now we get into validation. And there's a series of different cycles that you're going to do as part of that. You're going to do an empty chamber profile. This is where you're just testing the chamber by itself, just to make sure that it's operating according to its specifications. You're going to do a fractional cycle. You want to show that if samples have been exposed to ethylene oxide, that when you do the sterility test, then you're going to be able to recover those, and that sterility test would detect anything that might have survived. So that's your fractional cycle. Then you're going to do 3 half cycles. Again, you have to show that you kill those BIs in half the time. So you're going to have to do that 3 times to show repeatability. So you're doing it in triplicate. And then you're going to do either 1 to 3 full cycles. A minimum of 1 is required. This is where you test the residuals. Because ethylene oxide is a toxic process, you have to remove that from the product before you can use it on the patient. And so the residuals are done that. So again, the operational qualification, this is the empty chamber profile. This is just showing that without product in there, the sterilizer is acting like you think it should be, that it's performing the way you expect. And you're going to compare that temperature and humidity profile. Every year you look at that to make sure that the chamber performance isn't degrading. A lot of contract sterilizers will do a generic one for you, and then you can just get that data from them. They'll provide that to you, and then that just goes into your validation file each year. You do a sublethal cycle. Again, this is where you get some kill. It's what they call the fractional zone or the quantal zone. This is what you like to see. Hopefully you get some growth and some kill, because that way you can actually calculate exactly how much kill you've gotten on that process, and it gives you a point on that kill curve, and that helps verify the amount of lethality that you're getting. It's just one of the requirements from the standard that if you're doing that half cycle method where you have to kill it in half the time, that you also have to do a fractional cycle where you show you can get some recovery. So actually this is the one time that growth is a good thing. You want to see growth there. So then you do your half cycles. Again, this is where you're doing it in half the exposure time. So if you had normally a 6-hour exposure in your routine cycle, you're going to do a 3-hour exposure. This is where you prove that sterility assurance level. And it has to be done on your worst case situation. Really, whatever you validate, you're basically stuck with. And so if you want to do just the same load size every single time, then it's something that you're going to be basically stuck with that same load size. What some people want to do is they want to validate a range that gives them anywhere from a minimum range of maybe just 7 boxes, say, up to 10 pallets. And so you'll do half cycles on both sides of those ranges, from the minimum load as well as the maximum load. Find out which one of those is worst case, and then whichever one happens to be worst case, then you'll do 2 additional ones on that worst case situation. So at the end of the day, you have 3 half cycles on your worst case situation. So if you do have multiple loading configurations or different products that you want to mix together, you may do more than 3 half cycles, because you have to figure out which one's your worst case first, and then do 3 of whatever that happens to be your worst case situation. So then you do your full cycles. Again, the full cycle is just your routine production cycle. And what you're doing here is you're not only setting the ranges of the temperature and humidity, the physical parameters that you're going to go through from a normal production process. So once you get done with this, you're going to go off of those temperature and humidity ranges. But this is really where you find your residual testing, because again, it's a toxic product. You need to get that ethylene oxide off before it can be used. But you want to do your residual testing on samples that have gone through a full uninterrupted cycle, which is your longest worst case cycle. So that's where you do the residual testing. You don't do residual testing in half cycles, because that's only gone through half the exposure time. So you do it in the full cycle, and to make sure your product meets that. So when you're looking at your residual testing, it really depends on the contact that the device is going to have on the patient. Short-term contact, anything less than 24 hours. A lot of times a catheter might only be used for, say, an hour or 2 hours, falls into that category. Then you have prolonged contact devices. Those are devices that are used for 24 hours up to 30 days. And then you have implants, things that are permanent contact devices. Those are things that are meant to stay in basically for the rest of the patient's life. Then those are greater than 30 days, and then those all have different limits for that. So as you can see, the limits for the first 24 hours for the short-term contact are 4 and 9, 4 milligrams of ethylene oxide and 9 milligrams of ethylene chlorohydrin. Ethylene chlorohydrin is just a breakdown product of ethylene oxide. It's also a toxic compound. So then what you do with these prolonged and permanent contact devices, you have to do what's called an exhaustive extraction. That's where you keep extracting the device until you get all of the ethylene oxide off. If it's a short-term contact device, you don't have to do that. You're only concerned with, it's only used for 4 hours, you're only concerned about the amount that comes out to the patient during that short time, so you don't have to get all of it out at that point. But for the next 2 categories, you have to get all of it off to show what that total amount is, and if it's a prolonged contact device, you divide it by 30 days, and that gives you those limits. And if it's a lifetime contact, you divide by 25,000 days. So if you want to see how long you're going to live, see how close you are to that 25,000 days, because that's what they consider a lifespan. It's about 70 years is what 25,000 days are. So those are the limits on that. Another thing that they added to the standard was a thing called tolerable contact limit. This was not in the previous version of the residual standard. What this is it only applies to certain devices. It's only going to apply for skin contacting devices, something like maybe a surgical gown, something that's going to be on your skin that could possibly cause irritation. Because ethylene oxide can cause irritation, so they've set these other limits for skin contacting devices and implants that not only do you have to meet the limits that we showed you before, but if it's one of those 2 categories, you also have to meet these either 10 micrograms per centimeter squared or 5 milligrams for ethylene chlorohydrin per centimeter squared. So it's based on the surface area of the device. And that's just to prevent any localized irritation that if you have too high levels of ethylene oxide or ethylene chlorohydrin, that they could cause the patient to have irritation. So it's an additional requirement that was added. You also have a sample blank, and what the sample blank is, it's something that basically is not exposed to EO at all. You're just trying to show that when you do the residual testing that there's nothing on the device that's either going to inhibit the presence of ethylene oxide or enhance that. So you don't want anything that's going to mask that presence of that, so that would maybe be falsely identified as it. So it's really kind of a control just to show that when you do your residual testing that you know that really is ethylene oxide, the peaks that you'll get on the gas chromatograph. So that's the sample blank. The standard has, again, one of the advantages of ethylene oxide is you don't have to validate every single year. With radiation, you have to validate quarterly. Every quarter, you have to do another dose audit to ensure, because it's so dependent on your bioburden. The nice thing about ethylene oxide is you really truly only have to revalidate and do a half cycle every other year, as long as nothing's changed. You haven't changed your packaging, your product, the sterilization process, or maybe even the equipment. And so the standard has this nice decision tree that helps you go through that process. You assess what changes were made to the process and whether those were minor changes or major changes. If it's a minor change, and what you may do is just do a reduced qualification where you only do one half cycle. So instead of doing all those cycles that we showed you before, 3 half cycles, maybe 3 full cycles, with your requalification, if you've only made a minor change, you may only do 1 half cycle. Or best case scenario, if you haven't seen any changes, then what you'll do is you'll just do a paper justification, and you don't have to do an actual physical run. You just take all the data, you'll look at your bioburden data, you'll look at the history of any maybe non-conformances. If you had any failures during the year, maybe a biological indicator grew, you'll look at that, and you'll assess if you really need to do anything or not. In a lot of cases, it's just a written justification that you're documenting everything, that nothing's changed. So that makes it a really nice advantage of that. So that's kind of it as for a basis for ethylene oxide. Is there any questions about what kind of testing is required? And generally, we'll talk about this topic for 3 hours or 4 hours. So this is kind of a crash course in 45 minutes. So, yeah, right. Yeah, the question was, is there a requirement to inoculate either lumens multiple times or other sites? There's really not a lot of guidance of exactly how. In some cases, it says use the judgment of a sterilization specialist. But you're exactly right. A lot of times regulatory bodies, the FDA, notified bodies will question, why did you inoculate here, but you didn't inoculate here? And so it's a safer bet to inoculate more places than fewer. And then that way, generally, we'll inoculate sites that we know are going to drop off in the very first short fractional cycles. That way you have data for it. You don't have to try to justify it. Because anytime you justify anything, there's always going to be somebody that doesn't agree with you, and then you're not going to be able to convince. But there is no requirement that you have to do multiple sites along a lumen. I mean, we did a case where we did a closed end of a lumen and did the very far end where it was closed off, where the gas had to penetrate all the way down there. And we had a FDA auditor that said, well, how do you know the middle of the lumen wasn't harder than that spot? Even though it was fairly obvious because it was a closed system, it only could get in through the one end. They still required it to be done, and they had to redo that study and inoculate multiple locations. So just to get around that, we'll inoculate multiple locations along that, and then that way you don't have any questions. You have data to back it up on, even though you know that a lot of these are going to be a challenge. Although sometimes we'll find sites that we think are going to be easy to kill and actually turn out to be hard, or the other way around, you think they're going to be really hard and they turn out to be easy. So sometimes you're surprised by it. So it doesn't hurt to do more multiple locations on that. So good question. Thank you. All right. Well, that's it. Any other questions? Feel free. Yes. Great question. The question was for bioburden resistance. How often do you have to repeat that, or is that a one-time test? We mentioned your bioburden's always changing, so that's kind of the million-dollar question was, well, when do I have to do it over again? Because it's a test you really can't do one time and put it in the file and never have to do it again, because your bioburden's always changing. What the standard said is that you should repeat that at some periodic interval. That's not defined. It doesn't say every year. We recommend doing it annually if you're doing a recall. Every time you do a requalification, to repeat it then. Or if you have some unexplained or adverse events that happen with your bioburden, like suddenly you have a huge spike that you've never seen before, or your bioburden just starts for some unexplained reason going up and up, then you want to make sure that you assess it at that point to make sure none of these strange resistant organisms have been introduced into your process. So, but again, it's kind of up to your judgment of when you do that, not clearly defined.
And that's
ISO 11135, right? And that says there's a lot of different things in there that will say you have to prove that the resistance of the bioburden is less than the spore strips and things. There's several statements in there. It doesn't really tell you how to do it. It just says you have to do that. And then it's got to be repeated anytime there's a change and things like that. And so it's up to you as a manufacturer to kind of look at what changes have been made and then go from there.
You suggested quarterly bioburden testing.
Right.
Is that because 11135 doesn't
Right.
it's periodic?
Yeah, and what the question is, do I have to repeat bioburden quarterly? That's it used to be in the old version of 11135, it just said you had to establish what your bioburden was. Now it states that you have to go off of the bioburden standard, which is 11737, part 1. So it actually specifies you do have to do that document. That's more of a recommendation. I don't think even in that document it mandates you do it quarterly. That document was written more for radiation sterilization because it's so bioburden heavy. But that's generally what's done, because it's really more of a judge of your manufacturing process, and it really is part of your sterilization validation. So doing it quarterly allows you to, or at least multiple times during the year, allows you to see any seasonal variation, or anytime you add new people to your clean room, you fire somebody, you hire somebody else, they potentially may be bringing in new bioburden that you didn't have before. And so by doing it at some type of interval, again, there is some leeway for you to establish what that may be. The nice thing is once you characterize your bioburden and then drop that back, well, once you characterize it and show it's consistent, then you can reduce the number of samples and maybe even the frequency. Because again, with ethylene oxide, it's not a bioburden-based method. So, all right. Well, thank you so much for coming. Yes, sir. Yeah, the point here was that the location of your facility can also affect your bioburden. Is that correct? And so if you have multiple manufacturing sites, you may have totally different bioburden makeup as well as numbers on the product. So it's important to assess that if you have multiple sites or different locations. So good point. All right, we'll let you go to lunch. So thank you.